Hello guys and welcome back to Excelsior Academy. So previously we were talking about the cell signaling. We have discussed about the GPCR and tyrosine kinase, right? Let's just briefly summarize what we have discussed in the previous chapter, previous lecture about the RAF ERK kinase pathway. So that because the thing is that the other pathways which we are going to discuss today, it's a little bit related to these. So that's why just let us revise this thing. So once the growth factor comes and binds to the receptor, as you know, it is going to autophosphorylate, right? So this is going to phosphorylate this and this one is going to phosphorylate to this. So based on that, the SH2 will come and it will bind. So we also know that it is your SRC protein which has got the SH2 domain, which goes and binds to the phosphorylated tyrosine kinase domain. Then the JEF will get activated, will activate RAS GDP to RAS GTP, which activates your RAF. RAF is again a kinase which is going to activate MEK. MEK is going to activate ERK, and ERK phosphorylated goes inside the nucleus, or there it is going to activate the transcription factors and it is going to target the gene expression. Okay, now let's see the other additional pathways are there which are related to this. So there are the other MAP kinase pathways. So here, just for your reference, okay, so we have got the growth factors here. It can be any kind of cell stress or any inflammatory cytokines, which can activate your GTP binding proteins like RAS. The RAS की जगह पे यहाँ पे होगा आपका RAC, RO and CDC42. Okay, now this in turn is going to activate ASK1, MEK, MLK and TEC1. Now these are the proteins which we will be discussing during, cell cycle regulation. So please do remember that this is one of the pathway which we have discussed, and all these kinases are involved. Okay, so these pathways or these proteins will be discussed in the cell cycle. Then this in turn going to activate your the other MKK4, MKK7, again related to cell cycle only, and then Janus kinase pathway and the P38, eventually leading to the inflammation of the cell or the cell death. So what I want to show over here is that this is related to your MAP kinase. Okay, it is just that they are similar. Okay, now for example, same thing what we have just discussed now, like ERK, Janus kinase and P38 MAP kinase, it can translocate to the nucleus and phosphorylate transcription factors that regulate the gene expression. So there are multiple MAP kinase pathways, so they all function in all types of eukaryotic cells to control the cellular responses. The specificity of MAP kinase signaling is maintained at least in part by the organization of the components of each MAP kinase cascade as complexes that are associated with scaffold proteins which organize complexes of the signaling molecules. Now here I know that you didn't understand over here. What we are saying is that these MAP kinases, they are not present individually. Okay, they are again brought together with the help of the scaffold proteins. I'll show you the figure, then I guess it will be more clear. For example, the KSR scaffold protein, it organizes the ERK, RAF and MEK. So we know that RAF is activating MEK, MEK is activating ERK, right? So but all these, ऐसा नहीं है कि the RAF is waiting for MEK to come and then it is getting phosphorylated. No, the function of KSR scaffold protein is that to bring all these three different kinases together so that the upstream or the the downstream signaling can be passed nicely, or in the amplified version. So it is like this. So this KSR is the scaffold protein. Okay, so it has got the binding site for RAF, MEK and ERK. Now what is happening over here? All those signaling pathway downstream, it is happening. The JEF is activating your RAS, right? From RAS GDP, it is becoming RAS GTP. Now RAS is going to activate RAF by phosphorylating. तो अब ये यहीं पर ही है बाजू में. So RAF is going to phosphorylate MEK, MEK is phosphorylated to ERK, and then once ERK gets phosphorylated, there is a conformational change. It leaves the scaffold protein and enters the nucleus. Okay, now it is clear. So it is not that these proteins, in the cytoplasm waiting for them to come and so that they can get phosphorylated. No, because it has to be very fast. The signal has to be passed immediately within the microseconds range, right? So they are brought together with the help of the scaffold protein. Is it clear, everyone? Akshaya?
Yes, ma'am.
Yeah. So as a result of the specific association of these protein kinases on the ASR scaffold. Activation of the RAF by RAS leads to specific and efficient activation of MEG, and which in turn activates ERK. Okay, so following this, finally ERK dissociates from the KSR scaffold protein and it will go into the nucleus. So there are different scaffold proteins for different MAP kinases signaling. Okay, so this is how it is helping in maintaining the specificity of the signaling pathways inside the cell. Now the other pathway. So till now we have discussed GPCR, tyrosine kinase, and the MAP kinase pathway. This is your PI3 kinase or AKT pathway. So, why this happened like this? Okay, anyways. So it's a second major pathway of intracellular signaling downstream of tyrosine kinase. It is also stimulated by some G proteins based on the use of the secondary messenger, which is derived from the membrane phospholipid phosphatidyl inositol 4,5 bisphosphate, so PIP2. Now, we know that it is a part of the plasma membrane. If you remember, we have discussed about the phosphatidyl inositol or PI3-145 triphosphate. If you remember, in the plasma membrane, it is present. Okay, now, so PIP2, it's a component of the plasma membrane localized in the inner leaflet of the phospholipid bilayer. So of course, see, यहाँ पे कोई confusion नहीं आना चाहिए कि PIP2 आपका outer में कि inner में. Since the cell signaling is happening, so obviously it is going to get the signal from the tyrosine kinase or GPCR and downstream जाएगा, यानी कि towards the cytoplasm or towards the nucleus, right? So it is going to be present in the inner leaflet of the phospholipid bilayer. Now, it is phosphorylated on the third position of inositol by the enzyme phosphatidyl PI, that is 3 kinase. So, अभी क्या हो गया, ये आपका PI3 हो जाएगा, ठीक है? So, it will become 1,4,5 triphosphate now. So, the form of PI3 kinase activated by tyrosine kinase has SH2 domains and is recruited and activated by association with the receptor tyrosine kinases. So, here you have to remember or understand that once you get the signal goes, once there is a ligand or growth factor which goes and binds to your tyrosine kinase, right? So this is going to bind your receptor tyrosine kinases. Now from there, this phosphorylated, this is going to bind to the SH2 domains. Okay, now this is going to activate your PI3 kinase enzyme. Did you get it? I'll just repeat once again. So you have got the membrane. Here you have got your receptor tyrosine kinase. Okay, so we discussed already about the receptor tyrosine kinase. Now, the ligand comes, it binds, right? So what is happening then? The dimerization of the receptor tyrosine kinase is happening and they are getting phosphorylated. Now, this phosphorylated is going to get detected by the SH2 domains. Okay, this is going to then activate your PI3 kinase. A PI3 kinase क्या करेगा? It is going to add another phosphate group to the PIP2. Okay, which results in the formation of your PIP3. Okay. And this act as the secondary messenger. Clear? So in the previous, if you remember, in the case of the GPCR, we talked about the cyclic AMP. So their cyclic AMP formation was acting as the secondary messenger in the cell signaling. Now here we have got the PIP3. Yes? Chandani, is it clear? Krishita, Tanvi?
Yes, ma'am. Yes, ma'am.
Okay. See, the thing is, right now you have to remember all the cell signaling pathways, because later when we are going to talk about the cell cycle and the cancer, that time in detail we are not going. We will be just saying that because there is a mutation of this particular gene or this particular protein, that's why the tyrosine kinase pathway is hampered, or that's why the cancer is happening. Okay. For example, I'll say right now, like when we work. Okay, so as I mentioned before also that my main work is on the clinical trials. Okay, so I go through the clinical trials. So there directly they mention that TKI, that's all. What is TKI then? Tyrosine kinase inhibitor. So I should know tyrosine kinase signaling pathway so that I can understand this, how this inhibitor is going to inhibit TK inhibit tyrosine kinase so that it can prevent cancer growth. Okay, getting the point. So that's why at the back of your mind, you have to remember all the pathways, not only for your job, but also for the exam point of view. Okay, yeah. Now, so I hope this much is clear. So from tyrosine kinase, now we know how PI3 kinase is activated. Okay, now PI3 kinase is activated. Now this is going to add phosphate group to your PIP2, which is going to give you PIP3. Now this is important. Now the key target of PIP3 is a serine threonine kinase called AKT. So PIP3 binds to domain of AKT, which is known as plextrin homology domain. Do you remember plextrin, where we have talked about? Is it plextrin or it was something else that time? Plectrin. Oh, sorry, that was plectrin, not sorry. There's a difference. So this is plextrin homology. Okay, so it has got the pH domain which is present in the AKT. तो उसको क्या होगा आपका PIP3 bind करेगा. Okay, now this interaction will result in recruiting AKT to the inner face of the plasma membrane, where it is phosphorylated and activated by other two protein kinases, PDK1 and mTOR, which again contains the pH domains and binds to the PIP3. We'll come to this. Now here again, mTOR is also a part of another signaling pathway. We'll come to that together, and then please remember like how PI3 pathway and mTOR pathway is merging. Okay, please remember. So the formation of PIP3, it results in the association of your PDK1 and the mTOR, as well as AKT with the plasma membrane, which will result in the phosphorylation and activation of AKT. So once AKT is activated, it will phosphorylate many other target proteins, including the regulators of cell survival like bad proteins or the transcription factors. Okay, now let's see. We'll go through this pathway to understand how exactly this is happening. Okay, so here, as we mentioned before, so we have got the receptor tyrosine kinases. Okay, the growth factor comes and binds. Autophosphorylation is happening. You have got the SH2 domain binding to your phosphorylated tyrosine kinase domain. Now, that is the part. Okay, sorry. I said that this is going to activate PI3 kinase. Sorry, my mistake. So PI3 kinase has got the SH2 domain. Okay. Now, this gets activated. Now, this is going to activate your, it is going to add phosphate group to your PIP2, which is present in the inner leaflet of your plasma membrane. Okay, now as you can see, mTOR and the other proteins, they all are the complex mTOR and the PDK1, which is associated with PIP3 and PIP3. So all these PDK1 and mTOR, they get activated. They activate AKT. Okay, now once AKT is activated, it is going to have its role then. It is going to activate the further transcription factors. Clear? Don't worry about the bad proteins and the GSK3. We'll come to that slowly. Okay, is this much pathway is clear? Akshay, would you like to give the summary of this pathway? Okay, Chandani, would you like to try? Nobody? Krishita?
Ma'am, I was disconnected in between.
Okay, will you just try so that I will add, I'll say in between, no worries.
This I totally lost, ma'am. I couldn't
Okay.
attend.
No problem. Tanvi?
Ma'am, yeah.
Yes.
So, receptors are present in the plasma membrane, and growth factor comes and bind to it. Then it dimerizes, and then phosphorylation happens there. And to that, PI3 kinase, SH2 domain, it has SH2 domain, PI3 kinase, and this bind to phosphorylated end of that receptor. Then then PIP2 is present in the plasma membrane, and it get converted to extra phosphate is added, and it forms PIP3. That is mTOR. Then it forms mTOR C2 and PDK1. They both activate AKT, and then this AKT does his job of bad transcription factors and GSK3.
Okay. So it is mTOR C2 and PDK1. They get activated by binding to PIP3. Okay. So once they get activated, they activate AKT. This is this additional point. Good. Now it's clear. So how bad and how GSK is working, we'll come to that eventually, because there are many cell signaling pathways are there. So we'll come to that. So now it is clear like how PI3 kinase or AKT pathway is working. Is it clear? You will see that eventually all these pathways are merging together. So thoda bahut confusion aayega. So that's why you need to remember. Okay, there is a mTOR pathway itself differently. There you will see that this PI3 kinase or AKT pathway is also merging with the mTOR. Okay. Now the critical transcription factors. So here we were talking about, right, transcription factors. So one example is FOXO family. So the phosphorylation of FOXO by AKT creates a binding site for cytosolic chaperones like 14-3-3 proteins, which will sequester FOXO in an inactive form in the cytoplasm. Okay. See, this is how. So when there is no growth factor, so there is no cell signaling going on, right? So what will happen? The FOXO is free, so it can go inside the nucleus and induce FOXO-related genes. Okay. Is this much clear? 1 second. Yeah. Okay. So what it is saying that once there is no growth factor, FOXO is free and it can go inside. Now, when there is a growth factor, all this AKT pathway is taking place. AKT, the activated AKT is going to phosphorylate FOXO. So once it is phosphorylated, it has got the binding sites for the 14-3-3 proteins, and the FOXO-induced genes cannot be expressed that time. Okay? Now, what is 14-3-3? What is FOXO? Abhi yahi sawal aa raha hoga. Right? So just simple understanding is that FOXO is one of your transcription factor. Okay. And 14-3-3, it is your chaperone. Any doubts over here? Now coming to this FOXO later when we will talk about the insulin signaling, there also we will talk about it. Mainly this 14-3-3 or FOXO proteins, they are involved in the cellular metabolism. Okay, if a cell is undergoing oxidative stress, then only these proteins are getting activated. Okay, for time being, this much information is enough. Anything else you want to ask? Any doubts or regarding, like, do you want more details about these proteins? I can give, it's okay. So 14-3-3, now you know that it's a chaperone, so it will bind to the FOXO, right? Main function comes about the FOXO over here. So main function of FOXO will be just to balance, okay, between the cell survival or the cancer formation. Okay. So what mainly FOXO does is it ensures that your tumor suppressor genes are suppressed. Okay. If not, then what will happen? Cancer will happen, right? So they are they are present in the line which will decide whether the tumor suppressor genes needs to be suppressed or not, whether the cell needs to be proliferated or not. So all these decisions are done based on the expression of the FOXO. Okay, is this much clear? Yeah. Okay. So there are many cell pathways. Okay, cell signaling pathways are there. I am sure in your mind so many doubts were there, like why it is not making a connection. It will all make connections when we start doing the cell cycle as well and the cancer. Okay, don't worry about it. Now another target of AKT. So first is the FOXO, transcription factor now is the another protein kinase called GSK3, which regulates the metabolism as well as cell proliferation and survival. So like FOXO, GSK3 is inhibited by AKT phosphorylation. Now, any idea what is GSK3? Anyone? उसके full form से ही थोड़ा बहुत answer मिल जाता है. So what is GSK3? Any idea? Is it related to glucose? It is your glycogen synthase kinase. Okay. So obviously it is related to the, see when it says metabolism, so it is going to be related to your glucose or glycogen, whatever it is. So glycogen synthase kinase. So again, it is the AKT phosphorylation which is telling you whether this needs to be activated or inhibited depending on the requirement of the cell. Okay, so for cell proliferation, you need glucose, correct? So whether GSK should be activated or not, again, it is a depending on the growth factor binding, whether the AKT is getting activated or not, it is going to induce the gene expression. The targets of GSK3 include several transcription factors and the translation initiation factor, that is IF2B. Now, phosphorylation of eukaryotic initiation factor 2B, it leads to global downregulation of the translation initiation. So GSK3, it provides a link between the growth factor signaling and the control of cellular protein synthesis. Okay, let's see over here how it is being regulated. So once we have got good supply of resources, okay, in the first figure, please see in the first figure. So here we see that we have got the growth factor supply is there. Okay, so what is happening? What do we see? Your all amino acids are coming. Your IF2 is there. It is recruiting all these proteins, tRNA bound to your amino acids, and the translation is happening. Okay, since you have got all the resources available. This much is clear. Now, in order to continue the translation, your IF2, it needs to be recycled, right? GDP hai. If you remember initiation factor, they bring your new tRNAs. So IF2 GDP ho gaya. Now it has to be exchanged with the GTP so that can bring another amino acylated tRNA charged with the amino acids. Correct? If you remember the translation, wahan pe kya hota tha? IF2, it is bringing your new tRNA charged with amino acids. So after adding to the ribosomes, then they get released, hydrolysis hota hai. So IF2 आपका GDP हो गया है. अभी इसको दोबारा से नया tRNA लाना है with the new amino acids. So it needs to be recharged. So this recharging is done by your IF2B. Okay. So exchange of IF2 GDP with GTP. So this is happening in the case of normal when all the resources are available. Is this much is clear? Figure A. Yeah, okay. See, for that you need to remember the translation ka all initiation factors, okay, like how it is working over there or not. Okay, coming to the next figure, when you don't have proper amino acids or the nutrition available, or there is a stress or growth factor starvation is happening. One second, guys. Okay, now coming to this case, the second case when there is a stress and the growth factor starvation is there. Okay, so what will happen in this case? So we know that there are no amino acids available. So there are no new tRNAs with the amino acids, right? So what will happen that time? These regulatory proteins, okay, they will get phosphorylated and hence they won't be able to carry out their function. Did you get it? So EIF2B will be phosphorylated, which is going to phosphorylate your EIF2. So this is going to get blocked. So they are no longer functioning, because of which no new tRNA charged with amino acids will be brought. Hence, no translation. Clear? Am I clear?
Ma'am, can you just repeat the second one?
Okay, I'll repeat. Okay, so what we are saying is that here in the stress condition or when you don't have the nutrition proper, okay, nutrition nahi hai jada in the surrounding, you don't have the nutrition. So in short, no amino acids are present. Okay, now, so what will happen though the first amino acid has been added, let's say. Now this needs to get recycled, as we saw in this case, IF2 is getting recycled, that is getting charged with the GTP, and it goes and brings the new acylated tRNA, right? Here we don't have that. So in order to stop the translation, your AKT pathway will get activated, correct? It is going to regulate the downstream. So what is happening is that AKT is now going to phosphorylate your EIF2B, which is responsible for the exchange between the GDP to the GTP. Since it is phosphorylated, it is no longer functional. Okay. Hence, this is also non-functional now. It is also phosphorylated by the AKT. So what is happening? Because of these phosphorylated, there is a prevention of the exchange between the GDP and the GTP. Now, this is not recycled or not recharged with the new amino acylated tRNA. So nothing to add in the ribosome, and it is going to inhibit the translation.
Okay, yes, ma'am, understood.
Yeah. Okay. Okay. Now coming to the mTOR pathway. Okay. There are many pathways, guys. So you have to go through. I'll be sharing the video soon. So there are many pathways and they all are going to merge with each other. So don't get confused. Okay. So the mTOR pathway is a central regulator of cell growth and couples the control of protein synthesis to the availability of growth factors, nutrients, and the energy. Okay. So in short, mTOR pathway is responsible by in checking like whether the nutrition is available or not, should the cell divide or not. Okay, so that is done by your mTOR pathway. Now the regulation of mTOR is again by many multiple signals, also the PI3 kinase AKT pathway. So just now we saw that in order to activate your AKT protein, you need mTOR C to be get activated along with the PDK1. Right? So these two together activates your AKT. So that's why I was saying the mTOR pathway, the AKT pathway, they all are going to merge together later on. So the mTOR protein kinase, they exist in two distinct complexes in cells. So this is complex. If you remember in the previous figure, let me go back. Here, it is a complex. You can see the 4 to 5 subunits are present together, which is becoming a mTOR C2 complex. Okay. So we know that mTOR C2 complex is one of the protein kinase which phosphorylates and activates the AKT. Just now what we saw. So this is your PI3 kinase. Again, the repetition only just for your remembrance. So mTOR C2 is there, which comes and phosphorylates and activates the AKT. Okay. In contrast, there is mTOR C1 complex, which is activated downstream of AKT and functions to regulate protein synthesis. So mTOR pathway में आपके दो आते हैं अभी, ठीक है? पहला हो गया आपका mTOR C2, which is going to phosphorylate and activate AKT. ठीक है? Now second one is which comes downstreams of AKT, यानी कि इसके नीचे आएगा आपका mTOR C1. This is for your protein synthesis, whether to translate or not to translate. Clear? So this is your mTOR pathway. Now let's go to this. So there is a growth factor which comes and binds to the receptor. It is getting activated. यहाँ पे आपका mTOR C2 है, PDK1 है. It is going to activate AKT1, AKT. AKT gets phosphorylated and activated, and then downstream it is going to see when this mark is there, this dash is there, it means it is inhibiting. Okay, so this is inhibiting. Now let's say that this is low energy. So in the case of the low energy, your mTOR C1 complex gets activated. Getting the point over here? Low energy means low nutrition available. So mTOR C1 should get activated. For that, then we have got the AMPK. So here low energy is activating your AMPK, which activates your TSC, leading to the inhibition of your Reb, and this will result in the activation of your mTOR. So either you should have no translation or it should undergo autophagy. Now, why autophagy? So that the unwanted organelles or the proteins can be recycled for the survival of the cell. ठीक है, तो यहाँ पे अभी सेल आपका डिवाइड तो होगा ही नहीं, because you don't have energy, you don't have amino acids, correct? So it is not the question of dividing, but it is a matter of survival. So either they are going to remain like that or they will undergo autophagy. Okay, so that decision is being taken over here. Now, mTOR C1 is going to activate your X6 kinase, which is a part of your ribosomal protein S6, or it is going to inhibit the initiation factor. So eventually it is going to result in the inhibition of the translation. Okay, clear? Okay, everyone just go through this pathway once and let me know, are you able to get it? Any doubts over here? AMPK, it is your, I think, 5-adenosine monophosphate kinase.
Ma'am, what is this Rep and TSC?
Acha, so TSC, I mean, name wise it is different, okay? So because it was first found in the tuberculosis, so it is known as the tuberous sclerosis protein, tuberous sclerosis complex protein 1 and 2. Reb का full form नहीं है. I mean होगा, but I don't know about the Reb. So the full form here makes no sense actually, because see, many researchers were working on different proteins and जिसमें जो जैसा मिला, they have named like this. For example, okay, for example, mTOR. mTOR क्या हो गया? M is here for the mammalian. TOR is for the target of rapamycin. So they found that the rapamycin and an antibiotic is inhibiting the activity of this particular complex. So they named it as mammalian target of rapamycin, complex 1 and 2. So that's why I don't think anyone will tell you to go through with the full form, but it's okay, like for our understanding. Eventually what I have understood is कि cell bio में जो भी आपके पास ऐसे acronyms हैं, it makes no sense with the related pathway. Like for example, I have given you my example when I was working on the protein. So I have given the names to them. So depending on my understanding and my easy reference, so even I have given the names. BHL ho gaya, Dil9 ho gaya. So like that. So many cases are there. Only if you're reporting for the first time. It's not like that you are also working on TSC and you are giving some name to the TSC because you are working on something else. It's not like that, okay? Agar first time report ho raha hai in the world, first time you're reporting, then you can name according to your conventions. Clear? Okay. It will take time, guys. No worries. Take your time. I'll share this. What I want over here is that since these pathways are a little complicated, please go back, and of course I'm taking from the Cooper. You can go through different reference book also. Albert Bruce is also there. I do not recommend Lodish or Carp right now. because once the Cooper concepts are clear, which is given in very simple English, okay, once this is clear, you can go and refer Karp and Lordish. Now, when you should refer Lordish and Karp, once you're thorough with the concepts and the techniques used in order to identify this particular scenario or the test they are conducting, like how to understand this protein is present there or not. So what kind of fluorescence or what kind of confocal microscopy they must have used and why. Once you understand all these things, then you can go and refer Lordish and Karp. Okay, that's my personal advice. Otherwise, you can go through. I'm not saying those books are not good. Those books are the best. But for your understanding, I'm saying go first with the Cooper, understand it, the concept they are talking about, then refer these other textbooks. Okay. 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 So what just now we saw in this pathway, the description is given over here. Okay. So we are just, let's see the figure once again and let's go with those description given. So here we see till here it is clear to everyone, right? AKT is then blocking the TSC. Now AMPK1 is activating. Here it will be Reb, then mTORC1 gets activated. So either it is going to inhibit or carry out the translation or autophagy depending on the growth factor signaling they get. So mTORC1 is regulated by RAS related GTP protein Reb. Okay. Okay. Now which in turn regulated by the GTPase activating protein TSC. So here what you have to understand is that, see, Reb mein yahan pe GTP bound hai, right? So it is also another protein like your RAS. Okay. Now what is TSC? They are saying that it is a GTPase activating protein, which in turn regulated by the GTPase activating protein called TSC, tuberous sclerosis complex protein. Okay. Now AKT phosphorylates and inhibits TSC, leading to the activation of mTORC1 in response to growth factor stimulation. In addition, it is also regulated by another protein called AMPK. AMP is for adenosine monophosphate activated kinase. Now it senses, AMPK senses the energy state of the cell, and it is activated by a high ratio of AMP to ATP. So what do you mean by the high ratio of AMP to ATP? What is the energy state of the cell? What do you understand by this?
It is less energy state.
Correct. High energy means ATP hona chahiye, right? So when it is high ratio of AMP to ATP, it means the cell is weak. Okay, it is starving. Okay, having high ratio of ATP to AMP is then opposite. यानी कि बहुत ज़्यादा हमारे पास है, the cell can proliferate then. Right now it is under the starving condition. ठीक है? So AMP means weak है. Energy state is weak. Okay, good. So under these conditions, under low energy state, AMPK will phosphorylate and activate TSC, which will inhibit mTORC1 when cellular energy stores are depleted. So what is happening? AMPK is getting activated, okay, when the energy cell state is weak. AMPK is getting activated. It is phosphorylating and activating TSC. TSC क्या कर रहा है? mTORC1 को inhibit कर रहा है. ठीक है? Now, mTORC1 is also mTORC1 is also regulated by the amino acids availability, which are required for the mTORC1 activity. Okay. Now, mTORC1, when it is activated, it is going to phosphorylate at least two targets. What are those? S6 kinase and the EIF4E binding protein. Again, it is your initiation factor binding protein. Okay. Now, it controls S6 kinase controls translation by phosphorylating the ribosomal protein and the other proteins which are involved in translation regulation. And initiation factor 4E, it controls translation by interacting with the initiation factor EIF4E, which binds to the 5' cap of the mRNA. If you remember in the translation of eukaryotes, we discussed that 5 cap of mRNAs are being recognized by these initiation factors, okay, and the ribosome. So in dono ka alag se hota hai, and then they are brought together. Okay, so please remember यहाँ पे दो टारगेट हो गए आपके mTOR C1 के कौन कौन से S6 kinase and the initiation factor 4E which binds to the 5' cap of the mRNA. Now look at over here. So it is 4E which goes and binds to the 5' cap of your mRNA. Correct. So growth factor deprivation is there. So what will happen? Then the phosphorylation will happen of the 4EBP. This is 4E binding protein. Okay, 4E binding protein. So what will happen then? This is going to bind and then we are going to have the translation. Okay. So in the absence of the growth factors, translation is inhibited by the 4E BP binding protein, which binds with the 4E and blocks its interaction with the 4G. So you will see that if you remember like how in the translation all these proteins यहाँ पे तो सामने से दिखा दिए, but they have got the distinct binding sites. So they all come together and bind and interact with the 5' cap of the mRNA. Okay, now when there is a growth factor stimulation, it leads to the phosphorylation of your 4E binding protein, which will then dissociate with the 4E. So what will happen? Then this can interact with the A, B, and G, and the translation will happen. Clear? I'll repeat once again. See, when there is no growth factor, okay, growth factor नहीं है, translation नहीं हो रहा, क्यों? Because 4E binding protein is bound to the 4E, which will prevent its complex formation with the A, B and G. ठीक है, तो आपका ट्रांसलेशन नहीं हो रहा है. Now there is a growth factor stimulation, हमें मिल गया है. So what will happen? This mTOR C1 and all, they will come and they will phosphorylate your 4EBP. जिसकी वजह से क्या होगा? Conformation change होगा. It will lose its affinity for your 4E. Now it is free. Okay, 4EBP is free from 4E. This is free. Now it can interact with A, B, and G. So what will happen? Now your tRNA with the first amino acid will come and the translation will happen. Clear? Now it is clear? Same thing written over here. So in the absence of the mTOR C1 signaling, the non-phosphorylated will bind to 4E, inhibits the translation. When it is phosphorylated by the mTOR C1, it will prevent its interaction with the 4E. Finally, what will happen? This 4E is free to have interactions with the 4A, B, and G, and the translation will happen. Clear, guys? Yeah. Okay, so that's why I was always telling you, please remember your basics, translation, transcription, every time they will come. Okay, yeah. So in addition to stimulating protein synthesis, mTOR C1 inhibits the degradation of cellular proteins by regulating the autophagy. Okay, so we know autophagy is kya hota hai? It is the degradation of your cellular constituents within the cell. If suppose your mitochondria is not functional, so it will take the mitochondria and it will recycle the components. Okay, chahe wo aapka phospholipid ho, protein ho, whatever it is. Now when the cells are starving, so mTOR C1 activity is decreased, thereby stimulating autophagy and allowing the cells to degrade the non-essential proteins so that the amino acids can be reutilized. Clear now? Now is it clear? See, when the cell resources are less, AMPK will understand the ratio of AMP upon ATP. It will get activated. Okay. And accordingly, it is going with the pathway. तो आपको समझना है कि कहाँ पे कौन सी चीज हमें एक्टिवेट करनी है या नहीं करनी है. Clear? Yeah. That's all, guys. Any doubts?